Okay, good morning, everybody. It's a pleasure to introduce Dr. Annie Huang. This morning, Annie is a physician scientist and she works in a hospital for sick kids in Toronto. Her academic appointment is in the departments of pediatrics and lab medicine and pathobiology at the University of Toronto, and she was just promoted to full professor. Annie uses genomics to uncover new oncogenic pathways in rare brain tumors, and she had a hallmark paper a few years ago in Nature Genetics in which she identified altered epigenetic programming as a common defect in several independent embryonic brain cancers. And more recently, she has started to look into the spatial heterogeneity of medulloblastoma, and she holds 3 grants from the Canadian Institute of Health Research and is also a member of an international childhood astrocytoma genomics and epigenomics consortium. And last but not least, she is the PI on several multi-centered clinical trials. So I'm happy that you're here today and looking forward to your talk. Welcome.
Thing up. I passed that test. So thank you very much for this kind invitation. I was just saying that I hadn't been in Minnesota since I was a graduate student for a meeting a long time ago. So I thought today that I'd tell you a little bit about one of the projects that I do in my lab. And really the focus of my lab has been, since I started my program, to look at rare brain tumors of childhood, primarily motivated by the fact that in the clinic we're faced with the dilemma of a child with a diagnosis that nobody really knows what to do with or whether it's the right diagnosis. And so there was a space there when I started that almost 17 or 18 years ago. And so that's what I followed. And the idea is that, we started with from the bottom up. Since nothing was known, we sort of used genomics to figure out what's going on. And I have different programs that are at different stages. So we've gone past genomics into mechanisms in some, and some we're sort of still stuck at genomics. So by way of introduction, embryonal brain tumors is what I'm focused on. And for those of you who've heard many other people speak, this is the most common brain tumor category in children. So it's the number 1 tumor that you'll find in the very young to up to the adolescent age. And beyond this age range, generally, children tend to get tumors that are more like adulthood, so they tend to be more glial tumor. And within the embryonal brain tumor spectrum, it's a very age-dependent presentation. So I think we have yet to really appreciate the spectrum of tumor categories that can come at very different ages. So most common in this is the embryonal brain tumor is medulloblastoma, which is about 1/5 of all of the embryonal brain tumors. This is mainly my working space, where these are rare brain tumors, and there is really no real good epidemiological data, so this is guesswork saying they're somewhere between 3 to 5%. This tumor type has been much better described since a gene, an etiologic gene, was identified almost 20, 25 years ago. In this space are tumors that we are all fighting about as to what to call them. They're called CNS-PNET. I won't spend my talk talking about this. I think we still have yet to agree on what some of the papers mean and what we call them. But these are the major activities in my lab. Will suffice to say that within this space is, we've identified multiple different categories by looking at genomics. And I think the spectrum of tumors that has this label is expanding. And one of the ones that my lab has sort of gone deeper into is this category here that were named by multiple histological names, actually these 3. And we've shown that all these 3 categories are really the same molecular disease. Again, underscoring the power of genomics to try and get the fuzz out of some of the stuff that we do on a day-to-day basis in the clinic. So what is known about these baby brain tumors or these brain tumors is they generally arise, the rare brain tumors generally arise in younger kids. And I just wanted to put this up to show you what we make in terms of decision. These are grade 4 tumors, so they generally need something more than local therapy, and they have a propensity to metastasize, so from the primary site. And they generally look the same. They're what we call small round blue cell tumors of infancy. And there is sometimes not really specific distinguishing histopathological features that tell you they're different one from the other. In children, the approach is if you're greater than 3 years of age, this is a standard approach. You go from surgery, local treatment, to getting whole brain and spine radiation, trying to prevent metastases on top of chemotherapy. And this is the approach that's been used for all embryonal brain tumors. And it works very well for medulloblastoma, not so well for the other sort of brain tumors that look exactly like medulloblastoma, which is the rare embryonal brain tumors. And in this category, the age distribution is a challenge in delivering that kind of therapy. So the clinical challenge is that, they're very heterogeneous disease with unreliable diagnostic markers. We think they're the same, but they're not the same. And until recently, and it still is the case because there are no trials for some of these things, they're lumped together. And it's really, it's hard to define what the disease is, both from a biological and a clinical perspective, because we don't know what we're looking at and we're lumping them together in treatment. So maybe that's the reason why we have very poor outcome data. The majority of the children are very young, and from my perspective as a medical oncologist, it is actually the hardest thing to do, to decide what to do with these very young children, because we potentially could give curative treatment, but we know the consequence of that in a very short period of time. So we struggle with what to do, and there's lots of fights in clinical trials when we talk about what exactly should we do. We all have very strong opinions. So some of the biological challenges is because it's a rare disease. There are no real numbers. We don't even know what we're looking at. There were no disease models, and so that didn't really help us when I was starting this off. So one of the things that I did a long time ago, and it's now become more formalized because I now have a bigger lab and I have people who can help me do this, but I just went about collecting a lot of these tumors that people really had 2 or 3 of in their freezers or people talked about, and we formed a Rare Brain Tumor Consortium, which is now, I think, maybe up to 50 institutes that we are linked with, and we operate it out of our lab. And it's a web-based clinical database that people input clinical information to, as well as send us tissues to bank. And the whole idea is that we would collect these tissues and try and understand in an unbiased sort of way what these tumors are. And secondly, to sort of correlate it with clinical data, so we have some framework to start thinking about, what works and what doesn't work. So we've sort of used this to now sort of, in a very unbiased sort of way, go about asking, what are these tumors using the usual global tools? So we've gone from the first generation of gene expression arrays to SNP arrays to methylation arrays to the next gen sequencing tools such as whole genome, exome, and RNA-seq. And it's a very expensive endeavor, but you can imagine that we don't have a lot of tumors, so we have to go deep and figure out what's going on. We don't have the benefit of collecting a bazillion and figuring out what it is with one cheap tool, right? So we use very, very scrimpy methods for making sure we extend the life of a sample. So we use any material, and we develop techniques around that type of material that we receive. And more recently, we've sort of gone into the deeper aspects of asking, what is the functional genomics of these, and using epigenetic techniques. So this is just to give you a bird's eye view of where we come from. And my lab is born out of a question, and usually when we want to find out what the question is, we either look for good friends who have the technique, or we try to do it ourselves. So today I'll just tell you a little bit of the work that we've been doing in rhabdoid tumors, which is a tumor that has been long known to be associated with loss of the gene that encodes for this ubiquitous subunit in the SWI/SNF chromatin remodeling complex. So this gene is on chromosome 22, and the diagnostic feature is that you have biallelic loss of function alterations that takes away the protein completely. So this is like a BAF47 IHC, and really it's totally bland. And these little spots that you see are vascular endothelial cells that serve as the positive control. And it is a spectrum of tumors that are called rhabdoid tumors. The morphology is descriptive. The ones that arise in the brain are called atypical teratoid rhabdoid tumors. They can arise at other body sites, and are called malignant rhabdoid tumors. There are also many other tumors with this sort of a morphological description in the adult literature, and there's now some emerging evidence that they're probably, coming from the same place in terms of mechanisms. So these, about 1/3 or more of children who present with this tumor will have a heritable alteration of the SNF5 gene. And they can present with multiple tumors. And now that we've had a few survivors, I mean, sometimes now they have grown up and they actually truly present with cancer predisposition because they come in with a second malignancy. So children with the predisposition syndrome present early and with multiple tumor sites often. So my focus has been on the brain aspect of things because most of the tumors actually arise in the brain. The non-CNS tumors in children actually are a smaller number. So as I mentioned, these are rare tumors. It is the most common malignant tumor in infancy, which we define as less than 3 years of age. You can see that the median age of diagnosis is very, very young. We see children that are weeks old to 5. Beyond 5, there are rare kids. Most of them are less than 3. And historically, even when I started, it was considered a fatal disease. So it was a bad diagnosis, and we gave the talk that you can't do anything about it, and you either want to try it or you don't. And a lot has changed in the 17 years that we've been sort of focusing on this. First of all, people are beginning to recognize when they sort of do different things with intensification of treatment that this is now no longer a lethal disease. So I just sort of wanted to show you some papers that suggest now we have enough to say, these kids, some of them are living, and what are some of the prognostic factors. So this is an important study by one of my fellows, published in 2012, where we showed that even when you don't give them radiation, which is what everybody does as an autopilot, these children are living. So this was a very interesting clinical observation and bore the question of, what is the difference amongst children like these? They all have the same genetic disease, and yet they do very differently. So we have a couple of long-term survivors and a child who we walked away from. So we've always wanted to know, since this gene has been cloned and has lots of been studied for a long time, really, what is happening in this disease? So historically, people have shown that it's very characteristic. People have shown as well, Charles Robert Labs has done a lot of this work, that if you take away SMARCB1 in a mouse model, that's enough to form a rhabdoid tumor. Notably, that was not enough to form a brain tumor, suggesting that it requires a very specific niche. And more recently, people have done exome studies and said, there's nothing there. It's just this gene that's lost. And so the hypothesis came that these rhabdoid tumors may be epigenetically driven. These were relatively small studies of 14 to 15 tumors or so. And so when we were thinking about this, and we had been collecting for a while, we really wanted to understand, really, is there really no other thing there to find? And we started with the premise wanting to understand some of the clinical observations in heterogeneity, and then really hoping that we could sort of move from there to really developing models or biomarkers that would help us stratify patients or come up with new treatments, and certainly hoping that we can improve survival without causing harm. So one of the first studies that we did was low tech. So we used gene expression arrays, and so Jonathan Torkia, who graduated last year, and Daniel Picard, respectively, with a PhD and master's, were the people who worked on this project in the beginning. And we basically did transcription analysis, just saying, okay, we know they're genetically the same, but is there something different about them at the functional level? And so this is just to show you that when we did gene expression profiling, we were able to see a segregation into 2 different groups. And they were actually, signature-wise, quite different. So while one group looked very much brain-like, everything with brain was enriched, the next one was a little bit harder to pin down. And there was a lot of, a bone morphogenetic pathway signaling genes that were enriched. So we, by default, call these mesenchymal, the mesenchymal group, and we call these the neurogenic group. Excuse me. what was very interesting for us is that we saw very early on that there was some sort of a correlation with the anatomical location of the tumor with the signature. And that sort of suggested to us that maybe these were coming from different types of cells in the brain. So what we went on to ask was, does it matter? Like usually if you see something that's biologically significant, it should also usually correlate with some clinical significance. And so we asked whether this was one of those features that if you were neurogenic tumors, did you do better or worse than if you were a mesenchymal tumor or a childhood mesenchymal tumor? And so these are just two survival plots, just to show you that, you know, children who, regardless of the type of treatment they received, if they had the neurogenic tumors, did better. And so that was of interest to us because we felt like there was something to hang our hats on, that these differences that we saw were meaningful in some context. And so we went on to the next step. We wanted to ask, okay, so how does alteration of a single gene drive this clinical heterogeneity and biological heterogeneity? And you know, we and everybody else had been scratching their heads over this for a little while. And so we went, you know, because there were no models at that time of the brain, we did what we knew how to do, which was, again, to look at more tumors, but now using more wider catch techniques and deeper techniques to try and get to the bottom of why there would be this clinical heterogeneity. So I've listed everything on the slide that we've used. But basically, you're trying to sort of go back and forth between our genomics and going back to clinical association to really understand, you know, what is the molecular basis of the picture that we see. And so we've done, we went on to do whole genome sequencing because we felt that the whole exome studies didn't really ask the question that we did not cover the whole spectrum. So this is a circus map that shows all the copy number alterations in ATRTs, and there's about 70 tumors represented here. So it's a combination of whole genome sequencing and SNP array data. So basically what you see is this here tells you that all of them have chromosome 22 changes where SMART-B1 is located. But in general, it was a relatively sort of a bland tumor. But many of the coding, many of the alterations that we saw were not sort of classic point mutation sort of alterations. They were structural alterations. And that was probably part of the reason why people thought they were relatively silent. Having said that, it's only about 20% of tumors that have these structural alterations. And so that was something we found by looking at it a slightly different way. What we did see, which was consistent with other people's report, was that the coding alterations itself was relatively silent. So point mutations and indels and stuff like that were quite infrequent when you just look at the coding region. But what was really interesting was when John looked at the non-coding space, the mutation rate was 5 times higher. Again, sort of speaking to the observation by others that perhaps this is an epigenetically driven disease and that real business was outside of the coding region. So we haven't got very far with this. We're embarking on trying to, characterize what the non-coding alterations mean, but I'll show you a little bit of what we've done. So when we looked at this gene in more detail in larger number of tumors using more techniques, we were able to sort of see some things that weren't seen before. So in the old literature, it used to be thought that there was hotspot mutations between 7 to 9, and many people simply sequenced 7 to 9 to diagnose the disease. But what we found was that, in fact, there were no hotspot mutations. And the type of alterations, which was always thought to be point mutations, was actually in the minority. That much of the SMART-B1 gene is altered by structural alterations. So you have big chunky pieces of the gene missing. We saw some RNA fusion events. But what was really interesting for us was this observation that they didn't always truncate the protein, very early on. So you can imagine that if it sort of got this far, that much of the functional domains of the protein are probably still there and active at some point. And so when we saw this, and we haven't sort of drilled down in gory detail, but it made us think about the fact that maybe this isn't a pure loss of function disease. It might be a dysfunctional dysfunctional consequence of SMARCB1 loss. So we went on to do more of the profiling to try and figure out, are we seeing more now with larger numbers? And what we saw was now you can segregate things even farther. So our first group, which is very enriched in proneural signatures, stayed largely the same, and these the group 2s then now split into 2 further categories. And within each category, you can see that the signature is quite distinct. I think I put this up, and at the end you'll see why, is I think we never recognized this heterogeneity. And the more numbers we saw, the greater the heterogeneity. And perhaps we never really appreciated that the nuance of this disease that was only targeted by a single gene alteration. What was striking for us was that when you broke them up into groups, you could see that they actually were different genetic diseases because the patterns of loss, so this is a heat map showing the methylation group and the gene expression signature group, which largely correlates. But when we looked at the type of alterations within these groups on a global level, this group tended to be more silent. They often had sort of more focal alterations. And in general, when you looked at the extent of, I can't see very well here, sir, the extent of global changes, it was also different. In general, all of them had a very low mutation rate, but if you broke them up into subgroup, you now saw a different genomic spectrum. What was most striking was something that hadn't been seen before, was that the way in which SMARCB1 is lost is also differs amongst the groups. And so I sort of, that's why I was sort of saying to you that the way the mutation spectrum that we saw suggested that many of these alterations might not be a complete loss of function. And this suggests that it is dependent on the subtype, and perhaps that sort of determines what subtype of tumor you end up being. So we saw similar associations with location, and the association was much stronger. So if you were a group 1 tumor, you tended to have point mutations, and you were restricted largely to the supratentorial compartment of the brain. And if you were group 2, you were largely infratentorial. And if you were group 3, you were a mixture of infra, supra, and spinal lesions. And what was really interesting for me was this dichotomy or the spread in age. So if you look at it, the kids who are in group 1 are older. So this is the red peak here. They were all at least 2 years old. And then when you took the 2As, they were all babies, like they were here within the 6 to 12-month range. Whereas the group 2Bs had both a baby population and an older population. I think this sort of suggests to us that we were looking at perhaps a combination of what the type of genetic alteration was, where you got it, that determines the type of disease you're going to get. And this is what I meant by saying that in children, we have yet to understand the spectrum of some of the diseases because it changes with age. Your susceptibility to develop something changes with age. Part of it is because the disease develops in a moving target as organs develop in a child. So certainly for the brain, that's for sure. So one of the things that this sort of an observation brought was asking ourselves, does this mean anything biologically, and could it have therapeutic implications? The fact that you identify different types of patients, maybe they all behave the same in response to treatment. And there has been quite a bit done on SWE/SNF on the biochemical level, and there's been different papers that suggested there was some sort of a inhibitor for specific pathway that might be useful for rhabdoid tumors. And truly, if our observation was correct, one would suggest that maybe not all of these are going to act in one group, and perhaps there's diversity in it. And so that's what we wanted to prove. We wanted to say, okay, so we saw all of these different types, and really does it make sense, biological sense? So we went about doing a painful process because much as this disease has been described for a long time, people didn't collect cell lines. So it's funny because, we were Johnny come lately to this process and had the largest collection of cell lines because we really wanted to do this. So we collected a whole bunch of cell lines, generated some of our own, and then used this to ask the question, are the signaling features maintained in the cell lines that, phenocopy the features that we see in the tumors, and can we now use these reagents to ask whether there are actual biological differences and potentially therapeutic differences in the tumor subgroups. So this is basically just us doing a legwork to show that, group 1 cell lines were largely sort of driven or expressing Notch pathway proteins, while group 2 cell lines were not and largely expressing BMP pathway proteins, and that this was sort of largely mirroring what we see in primary tumor tissues when we did Western blot analyses. So starting with this, we then did the next step. We did a proof of principle screen and really asking the question, if they are different, they should be responding to different inhibitors of signaling pathways and drug or drug-like compounds differently. And we were a little bit surprised by this screen because it was really a proof of principle screen. What we saw was there was actually quite a bit of difference. And the group 1 tumors, were always more sensitive to a whole bunch of stuff, or cell lines were always more sensitive to a whole bunch of stuff. But the group 2s were much more resistant. And the thing that was most striking for the group 2s were these kinase inhibitors that showed up a lot. What was also interesting for us was that many of the epigenetic inhibitors that has been out there and that has been proposed to be important for SWI/SNF-related diseases were not sort of universally effective, suggesting against a nuance in the way these SWI/SNF complexes are taken out in each of these diseases. So that was really important for us because we had sort of thought that might be the case, but there was really never any proof that was the case. And this goes back to perhaps our observation that these children who have this tumor do better than children who have this tumor. So some of the, we went on to do the validation of the screen, and we were able to validate that, group 1 ATRT tumor cell lines were dependent on Notch signaling. So this is just an experiment where we showed, knockdown experiments using a generic inhibitor and then siRNA knockdown to show that when you actually took out Notch components, that the cells actually had an alteration in viability and growth. And that the reverse wasn't true if you took out Notch in a cell, in a group 2 cell line that we propose are largely driven by the BMP pathway. That didn't happen. And this is just a reverse experiment showing that if you take group 2 cell lines and treated them with DAPT, which is a loose Notch inhibitor, they didn't have much of an effect, so these red bars here. And that if you did a dose response curve for DM, which is dorsomorphin, that is targets the BMP pathway, you saw an effect. So we were quite happy that we actually were able to show that these gene expression maps that we see all the time meant something, and that indeed these groups of tumors were behaving in very different ways. So again, as I was indicating to you before, that the type of alterations you see of this SNF5 is different, the downstream effect and signaling pathway is different, their dependence on growth is different. So how does this happen when you take out one gene? And as I mentioned, the SWI/SNF chromatin remodeling complex has been proposed to be sort of the only dysfunction in this disease, and that this disease is solely driven by an epigenetic mechanism. And so we did some sort of correlative experiments to see whether some of the signaling changes could be explained by epigenetic changes. And we had sequenced a whole bunch, but we really did not find recurrent alterations to explain this enrichment of signaling pathways. So that's why we went to look at methylation differences between the different subgroups in the genes that are enriched within the subgroups. And this is just a heat map to show that whenever, in general, when you had hypomethylation of genes in the Notch pathway, that happened in group 1, and the reverse in the group 2 tumors. Underscoring the fact that probably it is largely an epigenetic disease, and things happen because the Sweet-Sniff complex is not able to function in regulation at different times in different ways. And this comes from the fact that, you know, a lot has been done in the yeast literature to understand what the Sweet-Sniff complex is. The Sweet-Sniff complex, I've simplistically shown it here, is a very complex, if you want to think about it. I don't think we yet realize how many different components come on and come off, and when they come on and come off. So a lot of the yeast literature has shown different components, but in multicellular organisms, I think the complexity has yet to be fully appreciated. What we do know is the fundamental mechanism is that, at least we understand it to date, is that this is a nucleosomal remodeling complex. So it allows the opening of chromatin regions because it's an ATPase complex that allows winding of chromatin regions and allows access of transcriptional activators or repressors or other regulatory proteins. So if you think about it this way, then you sort of wonder, like, how can you sort of prove that the fundamental difference between the different tumor types is in the way the chromatin is exposed, because that would be the reasoning. So we wanted to ask that question, and we collaborated with somebody who had this technique worked out to work on tumors. And so essentially it is a high resolution DNS1 hypersensitivity mapping, and it's the technique is called ATAC-seq. You're basically able to sort of sequence the exposed regions and generate a mega map of all the open chromatin regions. So we wanted to do that because our premise was that because the defect is thought to be in epigenomic regulation, that the patterns of open or closed chromatin should actually be different. So we, our collaborator helped us with this, and the first thing we did was we generated sort of these chromatin accessibility maps from primary tumors, and then asked the question, does this also segregate them? And this is just to show you that when you do what we call a principal component analyses, and we had a small number of tumors, but that they were separated based on the way their chromatin landscape looked. And this is just a different way of looking at it. And that was interesting for us because we hadn't sort of looked at it this way before. And what was when you drill down at this sort of data, what you can see is that many of the genes that we had, many of the genes that we had thought were important and upregulated, like the Notch signaling pathway genes and the BMP pathway signaling genes, when you drill down at the single gene level, you could see, based on these sorts of maps, that they were differentially open or closed depending on whether you're a group 1 tumor or a group 2 tumor. So I think this sort of allowed us to sort of think about it one more step. So these tumors are really, they're different at truly at the very fundamental level in the way their chromatin is packed. And we wanted to look at it a different way because it's very hard to prove that experimentally, but we have the benefit of looking at tumors. So we went to the tumors and said, okay, can we now look at it in primary tumors in a different way? And what are the sort of genes that are compacted and closed, and what are the genes are open? So we did something like this, which is, again, a summary, an informatics summary. We made a map of all the genes that were overexpressed in group 1 and that had corresponding open chromatin states. Now, just to sort of go back a little bit, group 1 tumors were the ones that had a neurogenic signature, suggesting that they were farther along in the differentiation program. So with a heat map like this, if you align the open chromatin regions with the type of genes that are expressed, so this is an RNA-seq heat map here, you see that all the group 2 genes are never open or in the group 1 tumors. And in the group 2b tumors, you similarly see, so these are closed chromatin, these are open chromatins when you see the red marks. But look at group 2a tumors. They're like open everywhere. So many of the genes are upregulated. And these are the baby tumors, as I call them. They have the youngest median age, and likely they have the most primitive features. We actually have data now in larger numbers to say that. And it speaks to perhaps an earlier progenitor cell of origin. So this is just a different way of representing that, showing the differences in the methylation level in the 3 different types of tumors. So SPSNF dysfunction in different tumor subtypes has a different effect on the way the whole chromatin landscape is regulated and compacted. As a consequence of that, the output of signaling is totally different. So one of the things that we wanted to sort of drill down a little bit was based on our observation that we saw this very different sensitivity to kinase inhibitors, and why, and what was the basis for that. And we did a lot of legwork to try and understand, as I mentioned before, through sequencing analyses, are they like conventional things that happen that lead to the upregulation of these genes? But we didn't see a lot. So we went on to use this sort of analysis to at least ask that question, whether it's true that the sensitivities of these different subgroups to different agents are also epigenetically driven. So when you take this sort of a data, you can do an analysis. I had a very talented graduate student, and you can sort of use this sort of chromatin openness data to now predict how different regions of the genome can act in trans or cis. So you're basically looking for nonlinear events, right? Could it be that epigenetic compacting or aberrant epigenetic compacting allows different regions of the genome to come together in a way that they weren't supposed to come? So you wouldn't need a mutation event. You would need just an aberrant epigenetic event. So when you do something like this, you can actually predict what might happen. And so we specifically asked, based on the fact that we saw that the PDGF receptor pathway was potentially specific to one subgroup, whether the PDGF locus or that pathway was somehow being epigenetically driven. So before we did that, we spent a lot of time convincing ourselves this was an important experiment to do. So we took the drug that we saw in our initial screen, which was dasatinib. We wanted to make sure that we really, really saw a subgroup specificity. And we did a whole bunch of cell lines over multiple experiments to show that while the group 1 cell lines were not at all sensitive, the group 2 cell lines were exquisitely sensitive. And just one step further, we put these, we treated mice with xenografted tumors with dasatinib, and we saw the sort of in vivo proof that we needed that this was really doing something for the tumor growth in terms of group 2 tumors. So as I mentioned, we thought the PDGF pathway was important because we looked up and down the yin-yang. It took us 2 years to figure this out. Like, why were these group 2 cells so dasatinib sensitive? And we went looking at every possible, pathway component. And at the end, we came back to PDGF receptor beta, even though we had passed it first thinking, oh, well, maybe it's not so exciting. But that was the thing that stuck out the most. But we looked very hard at our sequencing data, and we couldn't find anything that suggested the reason for this upregulation. And so we then did a series of experiments based on looking at ATAC-seq data to try and figure this out. So basically what we found was, this is a summary, is that in the PDGF receptor locus, there is the promoters down here. There is an enhancer down this way that was actually differentially methylated between group 1 and group 2 tumors. So we propose that probably the differential methylation of this allowed an interaction between the enhancer and the promoter in a cis, almost in a cis event. And that leads to the upregulation of PDGF, the receptor beta. So when you do this, with the ATAC-seq data, when you predict it, you can sort of superimpose that on some level deep epigenomics data to confirm that there is an enhancer there using HPK27 chips. And then when you do a detailed methylation map, you can see that there is as showing here, there's a difference between whether the methylation status of this enhancer element, depending on whether you're a group 1 tumor or a group 2 tumor. And this is just showing you that the neighboring gene, which we were just talking about, which is CSF1R, which we looked up and down the yin-yang a lot at, wasn't at all altered in any way, whereas PDGF was. And they're abutting genes. So this was a very specific event. And then we got help from our friends to show more directly that if you do an experiment like a 3C experiment, which allows you to assay whether these things actually come in proximity to each other in real life, that it does corroborate our observations using tissue analyses. And so these are just Western blot analysis to show when you treat these cell lines with dasatinib that it is the PDGF receptor beta protein that is differentially phosphorylated. So that was really interesting for us because I think it's a segue to what we think about the whole disease. And there are probably many other pathways that are altered in a similar sort of fashion. There was a recent paper which I didn't get a chance to capture by, actually, that's why I didn't capture it, because it's not published. But in any case, it sort of corroborates what we think, that this is not a mutation-driven event, and it is really a reflection of the way the epigenome is packaged in these different subtypes of disease. So this is just a summary of John's paper, where we showed that there are 3 different epigenetic categories of tumors, if you want to think about it, and that they respond differentially to treatment. So just to summarize, these are tumors that have different locations. They have different age distributions. The genotype of SMARCB1 is actually different, and the global genomic features are different. And as a consequence of these 2 events, the chromatin landscape is packaged differently. We believe this is largely because of the way, at least, it will take a lot of proving, but the data points to the fact that the event is probably very context and time dependent. So these tumors probably develop at a later stage in forebrain progenitors. These developed at an earlier stage in hindbrain progenitors. And it's hard to know exactly when these develop and what the cell of origin is. What we've shown is that as a consequence of these events, cells are either trapped or are able to drive very specific pathways to promote the transformation. And so these are Notch-driven pathways, and these are BMP-driven pathways. And as a consequence of these events, the tumor itself is also differentially sensitive to epigenetic inhibitors. So that's a bit of a, it doesn't come along intuitively, because when you think about it, the Swiss chromatin remodeling complex is a ubiquitous complex. So if you design an epigenetic inhibitor, you would think it should do the same thing for everything. But we didn't see that. And that was a little bit of a surprise for us. And how do you sort of think about it? And I'm not sure that it's really clear. So whatever I'm saying is all speculation. But so, around the time that we published our papers, others had seen similar sort of things, suggesting that, the ATRT subtypes seem to reflect different lineage derivation, mainly based on mapping enhancers. And many of these enhancers are lineage-associated enhancers. And so again, I go back to the question was, how does SNF5 loss affect different epigenomic consequences in ATRT subtypes? And it's a big question. And many people are working on it, and we are starting to try and attack it in different ways. But as I mentioned a while ago, perhaps it's not so surprising if you look at literature outside of our disease, because it's been known for a long time that Swiss sniff is very multi-complex and comes on in different ways. The switching of different complexes happens at different times during neural differentiation. And so perhaps it's not surprising because, you know, the Crabtree's group has particularly shown that during development, these switching events determine cell fate. And that is particularly in work from his lab has shown that the switching of between what we call progenitor or neural complexes happens in the course of brain development. And because we're looking at a disease that is arising during a developing brain, and perhaps that's why we get to see this, and we probably wouldn't see this in adults. So studying this rare disease has made us really think about a very simple concept, it seems, you know, homozygous loss of function is really not so simple. It isn't the dogma that I grew up with in graduate school, that it's a monogenic disease, you know, biallelic loss of function, and ta-da, right? It is really not that. It's probably different degrees of loss and different degrees of dysfunction that is resulting in the spectrum of phenotypes that we see. So this sort of just recapitulates that point, that if you think of this as being multiple components that can, you know, be changed, then this whole process will be a little bit different. And so we know from Crabtree's work that there are embryonic BRAF complexes, there are neural progenitor BRAF complexes, there are neural differentiated complexes, and probably, you know, our patients and their tumors are sitting on a continuum. And the effect of that, the loss, is felt differently, whether you're here, here, or here. Right? And so this is somewhat being shown by a recent paper from Charles Robert, where he shows that if you look at what happens when you knock down SNF5 and look at the enhancer landscape, the enhancer landscape changes. So suggesting that when you lose SNF5, the way the SNF5 complex normally sees the enhancer landscape in a normal developing cell is now altered. And that's probably what happens in our tumors. So again, this is just to summarize. So the loss of a single gene in this disease have different quantitative and qualitative aspects, which is sort of a little bit hard to get your head around a bit sometimes when you think about it, but not so hard when you look at what is already known in terms of the composition of this complex during development. So what we think is it's not only that you lose it, how much you lose, and this our observation of the genotype-phenotype correlation probably has biological and biochemical implications in terms of understanding what is happening at that time point when the cells are starting to be transformed. And so it's really hard to figure out how to attack this problem, because you can imagine, how do you generate, you know, just the right mutation, find the right window to target, and can you see the phenotype to prove that this is true? And I think that's the reason why it's been so hard for people to create a rhabdoid brain tumor model. So Charles Robert had shown a long time ago that if you took out SNF5, you can form tumors elsewhere, but he never was able to create a brain tumor model. And only recently has Frank Bordeaux at the Curie Institute, who spent 4 years making this mouse, was able to do that. So basically what he did was, sorry, I cut this off, he made a conditional model of SNF5 loss, and he was able to map only a very, very small window in which loss of SNF5 resulted in a brain tumor. So outside of that window, you could get tumors elsewhere, but you needed to be in this window to get a brain tumor. And what was really interesting was, depending on the window, he got slightly different tumors that recapitulated the features of the different subtypes in humans. So I think this actually now starts to make sense. And so now that we understand it's this way, how can we sort of think about therapies in SWE/SNF-related diseases? And you know, there's been a plethora of epigenetic drugs. How do we use it? When do we use it? And there's lots of trials now with these. And I think that if you choose to use it without selection, with the law of averages, you might not see a very profound result that you really, unless you really sort of focus on that area, you're not going to see it. So much as the same way when we start looking at subtypes, only when we looked at a very specific subtype were we able to appreciate that had these features that we didn't otherwise see if we took the whole group. So I just put this slide up because I think it's really important to think about that. Now, and studying rhabdoid tumors, I think, has made us appreciate this a little bit more. So it's known that the EZH2 PRC2, so PRC2 complex antagonizes Swiss function. This is known. And it's been shown by Charles Rabbit's lab that if you lose SNF5, that leads to upregulation or disinhibition of PRC2 activity, and there's as a consequence, there's increased activity of EZH2. So now there's lots of EZH2 inhibitors on the market, in the drug market. And based on our observation, I would suggest that the effect of EZH2 inhibition will be very dependent on SMARCB1 genotypes and the degree of Swiss function loss, the cell context in which this happens. And there's emerging data that it's not only the enzymatic function of EZH2 that's important, but the non-enzymatic functions of EZH2. So I think this will become an important area of work. And so for us, we're taking where we left off with all of our inhibitor screen and trying to understand, what kind of complexes are sitting in these different genes that define each of these different tumor categories by doing deep epigenomics. So we are multiply mapping. These are our primary tumors. We are creating models to perturb using small molecules, different components of these inhibitors, and asking the question, like, what is the consequence? And I think this is important because as we start to think about using these drugs, we need to understand, are they hitting the target? Is this what we're supposed to be seeing? Because if you are treating it in a context where you don't expect it at all, based on the biochemistry, why bother, right? So understanding what type and where the Swiss complex is sitting is going to be really important in terms of selecting who should get this treatment. Really, what we're asking is, what is the target epigenetic state you're going for? And that readout is actually a little bit difficult currently. Like, I'm not sure that we know how to read that out right now. But we're doing some of this work just to try and understand this. And it's beyond our disease, right? So what is really neat is, many of the children's diseases like retinoblastoma and Wilms tumor has helped us understand a whole bunch of biology. And I would say that similarly for rhabdoid tumors, which are uncommon tumors, but studying these diseases help us understand the entire biology between and the complexity of the Swiss complex. And this has implications for adults too, right? So because according to this review, at least 35 to 40% of adult diseases have some mutation of this complex. So starting with our studies in rhabdoid tumors and Charles Rabbit's work and multiple others, that interest that started with this very uncommon disease is going to inform much more common disease. So if you thought that was not complicated, well, it gets more complicated. So we have now collected over 200 tumors, and the more tumors we add, the more confusing it gets, and the more distressed your graduate students get, because they thought they had figured this out. So we've now gone into deeper interrogation and larger tumor sets. So we've banded together with some of the other groups, and now we see 5 subcategories. And what's really interesting for me is if you look really in detail, these 5 subcategories actually have very distinct clinical phenotypes. We now see even a much clearer picture of tumor locations. So for instance, here it's exclusively this tumor group, which we called the group 1, is now exclusively in the right frontal lobe. So you get down to precision like that. So now you start to understand, okay, what is it doing? Well, those are questions we want to ask. Why the right frontal lobe, right? So one of my research associates said to me, I noticed that they're all in the right frontal lobe. I don't have an answer why it is in the right frontal lobe, but it's very interesting. And I think I forgot to put the labels on, but this yellow group, which is the babies, are exclusively cerebral hemisphere tumors. So it's very cool to see this emerging in the patterns and mapping it. And so although, it has been a hard model to create in the mouse, some of the human data will now inform the generation of very specific models, subtype models. So what else can this do? One of the things that we've been really interested in to try and understand is what is the relationship of the brain versus the non-brain tumors. And I have a clinical fellow in my lab who wants to be a sarcoma person, so this malignant rhabdoid tumors are generally treated like sarcomas, and they don't do so well. And there's a bunch of sarcomas that arise in young adults that also don't do so well. So she started a project, and this slide is outdated because she's now got 70 tumors. We're starting to map these tumors in relation to our map of the brain rhabdoids, trying to understand, how do these diseases relate on the biological level, because we've actually been fairly successful in treating brain tumors. This is probably one of the areas that we have, in this area, in the rhabdoid space, we have a better survival than the non-CNS people, which is very unusual. So people are looking to us to ask, what should we do with the non-CNS ones? And by way of trying to address that question, we're starting to look at the tumors on the same plate. We're also very interested in asking, what is this non-coding space that we've seen, which Jonathan showed was being targeted 5 times higher than the coding space, and how does this contribute? And I think the PDGF story gives you an inkling about what that means and what are these. And so we actually are trying to do the deep epigenomics to map this back and understand what is the mutation sort of altering. We also know that there's a lot of stuff happening in chromosome 22. We've mapped this region down, and there are 5 or 6 candidates that are actually probably important modifier loci that contributes to the phenotype in this disease. And I think it's interesting, I speak very fast, but if you haven't caught what I said in the beginning, is that the genotype is very different. So in one group of tumors, the SMARCB1 is always lost with a whole bunch of other stuff. And that probably contributes to the phenotype in that group. So I've said a lot, and I think what I wanted to transmit was this, that by studying a very uncommon tumor, we've sort of have provided us, not specifically us, but the general community has provided insights into how a single loss of a single gene can have such pleiotropic consequences, and giving us insights into how just like conventional gene regulation, epigenetic regulation has multiple layers, and it's not going to be so simple to figure out how to target these tumors using epigenetic drugs. And I think the more we learn using systems and like ours that are natural tumors, the more we'll understand how to use these drugs. Because there's, for those of you who are in the clinic, I mean, every other day there's a trial in this, and who should you treat, and what are you expecting to happen? Right? So thank you very much for listening to me. I just wanted to credit the people who've done much of the work. So this is an old picture as usual, because I still had long hair. Jonathan was really instrumental in this. Daniel Picard is not on this picture. Polly Ho was a technician who helped with a lot of the cell line work. Mei Lu is a research associate in the lab who's also carried the load with doing many of the animal work. Alex Vasilovich was a neuropathologist from Lyon who spent a year in my lab, who helped with many of the pathology work. And Patrick Sinchan, who's a graduate student, helped with a lot of the cell biology. John was the informaticist, and Patrick has now gone on to work for Regeneron. So thank you very much. We wouldn't have been able to do anything without people sending us samples and being really interested in helping us solve this dilemma. So we have many, many more centers that are not listed here. We are supported by multiple grants, and really this project, if you can believe it, was started by the mother of this child who passed away at the age of 4. And she gave me $35,000. So, and that was, she passed away almost 12 years ago. And we remain very good friends, and she's been our strongest supporter. But she gave us the first $35,000. Okay, thank you.
Do we have any questions?
So now that you have these cell lines, I suppose you have the opportunity to try to re-express BAF47 and ask what happens. So I'm curious to know whether a subset of these epigenetic changes are reversible, and does the cell assume, depending on which group it is, a different differentiated identity? I mean, you're kind of implying that it's a very different target cell that suffers a mutation, and that's why you get different groups.
Yeah.
So I wonder if you'd see that in the cell lines.
So we've been trying to, so when you reconstitute SNF5, the automatic thing that cells does is they go into growth arrest. So you have to do it on a conditional basis. So it's a bit tricky. And some of the cell lines, they don't like behaving. So it's taken quite a bit of work to get that to go. And one of the things we've noticed that is, and these cell lines can only be sort of manipulated for certain passages. And after a while, they just don't look like what they were before. They're quite sensitive to culture conditions. So we have started to try and create PDXs and PDX models to do that. We've been somewhat successful. And so we're going to try and use that. And we've actually collaborated with I co-supervise a graduate student with Jim Retka, who's created an ATRT model. And we're creating now subgroups in our own way. And I think that reagent will really help us.
So, really nice talk.
Thank you.
How stable are these epigenetic states that defines the subgroups? The reason for asking, I guess the question is, if you were to do single cell sequencing, right? To what extent do you see rare populations within group 1 tumors that may be group 2?
Yeah. So you know, one of the things that happened was when we collected 500, what we noticed was there was more flux in the group 1 going into the group 2s. And I don't really know whether that's just a reflection of the way we think about, you know, our compartments. You know, it's a little bit of a gross thing to say supra versus infra, and when you look at the MRI a little bit in more detail, okay, and I get it, it's not really a supra infra. So I think some of these may have been mislabeled. But also, interestingly, what you were suggesting, so one of our patients, actually, we were just talking about it the other day, and we get a tumor sample. She had 2 metachronous lesions, one in the post-fossa and one in the super-10 compartment. And they came to the lab with different surgical labels, and people in the lab don't read surgical labels. They just know this is one tumor and the other tumor. And they were both profiled. Well, they end up in different space. So one is coming up as a group 1 tumor and the other one is a group 2 tumor. So I think there is some fluidity, I think, depending on the context.
It sounds like the pathogenesis, the tumor arising, is driven by rules of development, right? Certain things could happen, but once they get to a malignant stage, those rules may no longer apply because of the changes in landscape. The reason I asked the question is the fluidity between epigenetic states fundamentally dictates whether or not you could target.
Yeah.
Right? Although if you target one state to just escape into another state, you're not doing the patient any good. So that stability, or to what extent that is a stable state, is a fundamental question.
So I think if you look at the spectrum of tumors, so if you go from group 1 to 2As and 2Bs and now like the subcompartments of it, what I see is that the group 1 tumors are very differentiated. Like we have done deconvolution analyses of gene expression data, and we are moving to single cell sequencing. Even by eyeball, you look at the pathology. Like these are, you know, monotonous small round blue cell tumors in the group 1s. And if you merge yourself into the group 2s, you start to see classically the rhabdoid morphology, which is an expression of neuroepithelial morphology, mesenchymal morphology, and so that's where the teratoid label comes in. So I think that sort of group of tumors probably arose, at least this is my speculation, a much earlier time where there's greater plasticity in the epigenomic landscape. And so those ones you'll see greater heterogeneity at the single cell sequencing level. My bias is that the tumors that are arising in older children that are neurogenic, that have neurogenic signatures, will have less plasticity. So we're starting to embark on single cell sequencing. We haven't done a lot of it. We're just sort of trying to get the technique to work. because we have to get it to work out of frozen tissue since there's not a lot of these tumors. They come frozen to us, so we have to figure out a way to look at them. We don't have the benefit of having viable tissue come to us. So what we see based on deconvolution data, which I didn't show, so one of my graduate students is doing this, taking all of our old gene expression RNA-seq data and asking the question, if I superimpose known gene expression data sets, what do you see? So we see that. We see that the group 1s are really looking like brain cells. There's hardly any other cell types there. And the other ones, it gets busier and busier as you get to the younger kids. So that might be the reason why. I think it might be that it is a more plastic epigenomic state when you start with the earlier progenitor.
So with the changes in the chromatin landscape, have you looked for any neoantigens that might be suitable targets for immunotherapy?
So that's what people are doing, and I think that we haven't. There is lots of people who are interested, but I do think that makes sense. Because if you look also, now that we have more tumors, and we haven't published this data because we're still looking at it, again, the space in which the younger kids come in, their SparkB1 genotypes, as I was mentioning, takes out a whole bunch of other genes, and that speaks to genomic instability as some part of their signature. I think if anybody is going to get that, it's probably this group of patients. And in our deconvolution experiments, we smell a lot of immune modulatory pathways. Yeah.
Thank you.
Yeah.
So speaking of genomic instability, I'm just wondering whether within these defined developmental windows, there are changes in the replication program of these different precursor cells, whether there's anything known about it, because replication problems are normally the number one cause for these structural rearrangements.
Mm-hmm. So I think one of the things we don't know, so the literature suggests that 35% of these kids will have a germline alteration of SNF5, and that's the beginning. And there's controversy as to whether loss of SNF5 actually results in genomic instability or not. And we've sequenced a fair amount, and we haven't seen a lot of other alteration, but we haven't sequenced every single one, the germline of any one of these kids. There are a couple of kids on the right hand of the 2As and 2Bs that have other alterations that are in DNA repair pathways. And I think that in those kids, I actually think the SNF5 is a secondary consequence of some sort of a genomic instability event. That's what I think. I don't have enough data. It's a one-off, 1.5 kid, you can't talk about it, but that's the gestalt of the data. Yeah.
So you quite elegantly showed the differences between the different subtypes and the epigenetic basis for those differences. I'm really curious whether you think there are commonalities between the rhabdoid tumors and, say, a normal developing state. Whether you think that the landscape, the developmental landscape, drives very different effects of SNF loss, or whether we just haven't revealed the commonalities that are present because we don't have really good sort of normal developmental states to see those differences.
Mm-hmm. I think you're right. I think, part of the struggle with studying embryonal tumors has always been, what's your normal control? What window are you looking at? So I think we don't know what we don't know, right? Sounds stupid to say that, but it's true. Because I don't know that we truly understand what the composition is during development. Much of the work has been done in yeast. There has been mouse work done. One of the things that I think is really intriguing for me is some of the things that we think happens that's known, that's been proven in yeast and in mice, that we see, like the expression of what are supposedly microRNAs that switch the subunits around so that they become at different stages. And maybe that's a more stable event than the Smeesnov complex. So I don't really know. One of the things that we really want to do, and I don't know whether that really answers that question, is to capture the complexes in a biochemical state at different time points, right? Because I think a lot of what I've shown is an association study. It isn't like, it's just showing you genes that come together. Like, we have no way of knowing what the true biochemical composition of these things are. And those have never been done in different cell types, normal cell types, neural cell types, nor in a disease cell type. And I think maybe those sorts of experiments might help us address sort of, is there a common part to this that we're not seeing through this sort of study? Do you know what I mean? I think it's a difficult question. It's very hard when you're looking at this data, because you're sometimes like, okay, like, what is the normal counterpart? What is normal? What is abnormal? And we rely on we're constantly scouring for normal data sets, constantly. Like every other day, somebody's gone on, okay, who is publishing another data set of normal just to compare. But I know that there are several initiatives where people are actually dissecting different normal brain, and one of them is actually at our local institute, and they're generating single cell deep epigenomic data, and those sorts of data sets I think might be able to address some of these questions.